Okay, it's all yours, Gustav.
Yes, thank you. Hello to everyone first. Um, we have a good round together. Um, Let me make a couple of remarks before I start. It will not take too long, but I do have to make a few perhaps personal remarks. What follows could be could be seen as a little biography, um, a kind of, but it shouldn't be a biography. It was, I think, Ken himself suggested that I would perhaps a little bit engage in my previous background that led me to consciousness studies. I think it's quite a good idea, but as I say, I do not want to be too self-centered on these issues. But I think there is a line in my previous work that led me, for me, quite convincingly to the issue of consciousness. Another remark I have to make shortly, it's a kind of personalized issue. So, but I remember that we had some uh, discussion once about the lack of personality in science. So I would like to bring it in again. I think our more recent development, for instance, in artificial intelligence, kind of starts to depersonalize science a bit much, but it's all made per persons. And I do have to add another point. I'm not a specialist in all these disciplines that come together in consciousness studies. You get high technical specialist people. That's one of the issue that if you engage in consciousness studies, you have to generalize and you have to sacrifice some of the uh, disciplines to be a specialist in particular disciplines. But on the other hand, you get some more insight in diverse fields. I'm not a very good discussant either. There are several reasons. I have a hearing problem myself, and the computer here has another hearing problem too. It's um, so, uh, sometimes I have to use my headphones to understand questions, and some other times I lose my own voice there. And I like to think about questions anyway, so I'm not very good responsive to discussions immediately. I like to think about things first. I have about 20 slides here, but I could drop out about 5 slides that are engaging in a kind of case study that I could as well just summarize very quickly. Uh, all these remarks, I wouldn't advise any young scientist to make these remarks before he starts a talk in front of an audience, uh, because there are a number of apologies. But anyway, I did want to mention this, especially during these difficult times that we have right now. Anyway, Ken suggested, well, I have a problem in. So, have to go back. I split it up in two parts with the second part following next week. I think it's a good idea because I usually stuff too much into one talk. And so it lets me talk a little bit more loosely about these things. The part one today will be my personal landmarks and supportive background for these studies. I think they're always important. And next week, we will become a little bit more precise about what is at the center of my attention. This is the mirror sense transformation. realized by the brain. And the claim is that this sense transformation actually cause conscious transitions. They're not just correlating, but causing them. Uh, okay, now this all is a part of a little, well, extended manuscript right now that I tend perhaps one of these days publish in the form of a book, but it's not finished yet. Anyway, on the right side, I usually give some citations. I have to shift my uh, my points up there, not to get confused. I don't know how to do it right now. Anyway, okay.
Are you worried? Is it because you're covering up some of your slide? You can move that little window that shows you. You can move it way down into the bottom,
Okay.
and it'll be out of the way. We don't see that as the audience. We see your whole slide, but I know when you're speaking, that little window sometimes is in your way.
I might do it. I'm not under control right now of this little window that you mentioned, but I could come up with it. Anyway, part one should uh, dealing with this personal landmarks on this route to consciousness. In my abstract, I suggested very similar issues to Ken's tripartite proposals about three levels, with some differences that we might eventually agree on. Uh, the level one would be what Ken called a machine robotic life. I would call a primordial life. It's because life must be there. It's self-sentient, self-replicating, and it builds on energy transducing membranes. I will come back to this point a little bit later on. Preconscious life forms are the intermediate level two. They are equipped with a signaling plasma membrane that comes on as soon as we proceed to eukaryotic cells. From prokaryotic cells to eukaryotic cells, we internalize the energy transducing membranes and develop and make the cell membrane, the plasma membrane, free for signaling purposes. I think that's good for level two. And finally, level three is the conscious form with a nervous system. So pre-conscious life would, for instance, also involve plants. You have slightly ignored plants. But they cannot become conscious, and level three can be conscious, but doesn't have to become conscious, and it needs a nervous system, electrically excitable cells there. I've surrounded these three levels, which are epistemic levels, accessible to experimentation in principle, with two rather speculative ontologies where the origins are there in the prebiotics arrangement of atoms. And that's another deeply, intensively discussed issue at the moment. But there are some assumptions there that we could eventually come back again later on. And finally, another level that follows from the three levels is a kind of mentalization, becoming independent, accessible to speculation, of course, but I would just at the moment call it the disembodiment of things, where the mental experiences kind of develop their own, maybe uh, independent existence. So this opposes somehow very much the an idealistic view. It opposes very much also a dualistic view. If the dualism sets out from an independently existing mental world and a physical world that combines in the organism, but it just says it everything develops from the organism. It's an empiricist view. You know the big names like the philosophers behind empiricism. Uh, it says that all thoughts, concepts, and ideas are emerging from our sensory perception. And I'm quite convinced that this is a plausible way to see it. It emerges from our sensory perception from the three levels, and the three levels are mutually dependent on each other. So level one builds on, or level two builds on level one, and level three builds on level two. They are conditional, preceding levels there. So anyway, uh, little sketch from this. I distinguish between different kind of addressable uh, conditions or spaces, if you want. There's a subject. It's in the middle of, in the center of our consideration. There's an object place and a kind of physical place. There's a brain space, also a physical space that we can investigate. And there's the subject's experience D. And sensory experience leads eventually to concepts, as I said from above. The transition, how sensory meets concepts, occurs in the brain. And the question is, what is this transition? And where's consciousness there? Because it can be there or cannot be there at level three. Because it's a transient issue, consciousness that builds on the same organization of level three things of the nervous system. The nervous system does not reorganize when we have a conscious experience or change this experience. When we look around, we change the experience, but we hardly have any, let's say, essential synaptic uh, changes or whatever in the nervous system. It's one and the same organization giving us access to an enormous range of experience, conscious experience. Carl Friston was one of the persons, I did not cite him yet in this line. Carl Friston was one of the persons who wrote up a larger article in Science once about this point. Now, anyway, the mirror metaphor is essential for my point, and I'm taking it quite literally. On the right side, you see that Robert and Susanne Mice, who are with us today, hello, uh, suggested this point as well recently in a talk, and I think it's an excellent metaphor. A metaphor use a different reference to express something. So the reference doesn't have to be taken literally as such, but I am very much in it. But it tells us a lot because it tells us we have to manipulate ourselves to have conscious perception. And that self-manipulation is physical. So that is, if this is true, then the cause of consciousness is also physical. And it's inside and with the brain. Perhaps unknown, or there are different aspects where we have heard different suggestions for it. They may all apply, but as a matter of fact, what they have in common is that they're a physical cause of consciousness, building on level three. The mirror is a forward to back transformation. And in the course of that, because we point our nose towards the mirror and the mirror points his nose towards us. And in the course of that reversal, we have an asymmetry between left and right as well, which is quite funny sometimes if you try it out. But you don't see any mirror if you don't light on yourself, as Robert pointed out last time. And that's the issue of a self-manipulation. So some of my talk today, I think you noticed it anyway, will remain fairly general, but subsequently, I think we will go into quite a few details. Uh, Now, let me uh, then explain where is a continuous line in a set of rather diverse studies that I have performed during the last decades of my active time. And I think it can be found in the principle of invariance, which is well known in physics. And it goes all the way through, and I will need it to explain the final issue of consciousness very much. Now, let me explain this invariance principle roughly. We will not go into any deeper into mathematical details, except a couple of chapters perhaps next time, but uh, stay very general in a kind of, ordinary first-class mathematical issue. Felix Klein from Erlangen's program, I think it's quite intuitive, his definition of geometry. And I very much adopt a geometrical approach to all of these questions. I think geometry is enormously inspiring and a groundbreaking basic of science altogether. And the invariance, he defines geometry in the study of invariance on a particular set of transformations. So he characterizes and distinguishes geometries with the help of group transformations. So that the pair of, we need an object, that's the space X, and the group transformation G there. And it's a mapping of the pair G and X into the space, staying within that space. And a property is G invariant if it doesn't change anything. Let's put it that way. If it doesn't change anything. Now, the group operation here will be multiplication. It's a group. It's the group of multiplication within a standard element, and this is 1. If you have a group that builds on addition, this element will be 0, and it doesn't change anything. And then it is G invariant there. And I give a couple of this transformation on the left side. Euclidean geometry would be invariant with rotations and translations, preserving shape. Affine geometry would preserve collinearity. It can change shape, but in a collinear way, it doesn't change squares into circles. And topology is the more general issue. Topology studies shapes that remain invariant under continuous manipulation and changes. The shape doesn't play a role at all. Then finally, we have the sense relation in blue here, shown with very simple elements that builds on a certain asymmetry of the element. If this is a one-dimensional element there, this would be its mirror image.
Yeah. Here's the mirror.
You see that a transformation of these two simple things is not very easy at all. Actually, we have an embedding space. The embedding space for this one-dimensional object here is the plane, it's 2 dimensions. And within that 2 dimensions, without secting it up or dissections, you cannot easily perform that sense transformation at all. You would have to use a third one, if another helping, assisting dimension. you can use the third dimension. Then you can simply rotate this object around, giving that one. But staying within the same dimension, you cannot easily perform it unless you section the objects, like is done in chiral chemistries very frequently during the last 10 years, and there have been even 1 or 2 Nobel Prizes for this. But we don't section these things. We just look at the sensory relation as such and see how it can be done eventually. Now, there are different kind of views behind the principle of invariance, because the mathematical issue, for instance, I just explained with Klein's world, or Felix Klein's interpretation of geometry. And every physicist knows the importance of invariance, of course, in physics. Physics, actually, the structure of physics builds on the principle of invariance very much. But there are other applications, other lines of invariance. The physical invariance builds on symmetry groups in special and general relativity, for instance, local gauge symmetries. Noether's theorem, you talked about this a lot, conservation laws are following from this. Psychological invariance is a little bit less known. I have to shift this a bit down. It's a little bit less known. It's psychological invariance studies, for instance, projective geometries and visual perception, where the invariance in projective geometry is another issue. It's a cross ratio. It's the invariant there. And there is something like sensomotor invariance in vision. Now, these chapters have been reported in many papers, but they are kind of forgotten. One of the leading persons was Suppas from Stanford University in the 50s and 60s, the principle of invariance with special reference to perception. There are biological invariances depending on the scaling of organization. That's at the center of my attention in this work. And one of the issues about this biological invariance and psychological invariance I studied before, and it's given in this paper. I will come back to this paper in a minute. So anyway, the principle of invariance is basically a mathematical one. It's got the structure in physics, defining many things in physics, but it's got application in psychology and biology. And I will focus, of course, on the psychological and biological invariances in this work. Now, giving you an example of psychological invariance, this has been well studied. One of the more famous work is from Charles Calistel. It's impressive. At the end of the 80s, he came up with a book on the geometric module in the red. It's quite interesting. And he performs this kind of transformations that we talked about between affine or Euclidean affine geometries. He performs it in the maze, performing behavioral studies, for instance, studying the rat, seeking for food at different locations with a marked properties. And he studies the displacement of these corners and the geometry behind it. Displacing all corners, for instance, counterclockwise, is the same as a two-step affine transformation followed by a rotation. And the question in his experiments is, can the rat still perform finding its food location as quickly as before, after the transformation? that's a kind of
behind it.
studying psychological invariance principle in experiment. Okay, now influential landmarks for my consideration. I want to mark them in 4 steps. I have invariances studied in neuroscience, behavioral biology, I just gave an example, eventually in neurodynamics, and eventually in neurophysics, modeling and simulation from classical to quantum scales, which will be at the center during the next talk, part 2. Now, neuroscience was the beginning of my work in measuring the brain at different scales, and I'm giving a couple of examples there. At the large scale, reconstruction of whole brains, measuring brains, volumes, shapes, down to the cellular level, the tripartite system. I'm quite proud of some of these images once because that's a silver staining, and everything I show comes from my own work. And that's a silver staining, which shows us 3 partners in the brain that cooperate very closely. It shows us a neuron here, a pyramidal neuron from layer 3, actually, a blood vessel here, and glial cells, and astrocytes, and fibrous astrocytes. And the cooperate, there's an inhibitory cell there. With a different dendritic pattern. Here we have the oriented dendritic pattern. But this tripartite system explains a lot of function in its cooperation as well. I'm not going into details. We studied a lot of cell patterning in the brain. For instance, studied the tessellations, Voronoi tessellations between neurons and glial cells in the avian brain. It's from the hypopallium, the most dorsal part of the avian brain. Looking whether the patterning makes a lot of difference to the function of the system. And it's a composition of different immunologically staining the cells. The neurons are in blue and the glial cells are in red. There are many more glial cells than neurons. And they range in certain patterns, but it's the issue is somehow a little bit different. Now, this kind of work has been studied and right from the beginning as a young postdoc. I started with the foundation of computational geometry there. Then I studied the cell patterns during sexual neurogenesis, which is driven by hormones. At the time where, where the sexually dimorphic brain studies came up very importantly there, basically concerning the hypothalamus, the sexual dimorphic area of the anterior hypothalamus. Eventually, with the issue of avian behavior, comparing migratory birds to sedentary birds, whether they have different brains, either volumes or patterning. And then and there are a number of papers there where this is all published. Let me summarize the essential part now, because as I say, there is a line going through invariant principles, and what came out from me is that topological invariance, invariants that come up under continuous deformations of shapes, such as number and connectedness, are important for the level 3 organization, more important than patterning somehow. We are a little bit too much concentrated about the layered the layered or laminated cortex in mammals as compared to birds that have an aggregated forebrain, but with very much the same function. So it's not really all that important how you arrange the cells, but number is a very simple topological property is very important there. The numbers and the connectedness. And there are an increasing number of papers coming out right now. I will cite them next time that discuss these topological properties in the analysis of the brain there. Also, with respect to perception. Now, psychological invariances, opposed to structural ones, is measuring behavior, is mirror studies, spatial behavior. I engaged a lot in the behavior of animals in front of mirrors, studying precocial birds or altricial birds, precocial birds like chicken or quail in this case, using preference map studies where the behavior, the motor behavior of the chick is recorded together with its vocalization in front of a mirror. It's again something like a polarized maze, as we have seen before in the rat. Polarized because the maze, that area of the square has different meanings, and the different meanings are imposed by some object. In this case, it's a box with a mirror. Okay. In course of this, I find that there is a behavioral invariance principle again. And that principle of invariance is the seeking behavior of sensory invariance-driven action. That means the subject tries to maintain some important sensory input by a kind of feedback relation, sensory-driven action feedback relation, from its internal representation to its location in space, in physical space, in the maze, tries to maintain it. And that's quite similar to adjusting, for instance, orientation, visual orientations in lines to the configuration of receptive fields. That issue is a little bit complicated, so I'm citing my cooperation on these issues with Jaak Panksepp. I think for the US psychologist, he is very well known because next to Joseph Ledoux from New York, Joseph Ledoux has been mentioned several times in our sessions as well. Jaak was one of the initiators of affective neuroscience. And I cooperated with Jaak for several years and was a visiting professor in his lab there. Some of the studies are taken together in this work and came out in Neuroscience and Biobehavioral Reviews. Also, we joined the studies with molecular components and found out that the distribution and numbers again of opioid receptors is strongly responsible for this kind of behavior. Okay, that goes back quite a while, but as a summarize here, behavioral invariances are there. Subject is seeking sometimes to maintain a preferential input, let's put it that way. And that's important to experience it. Okay. Psychological invariance measuring behavior again, another example. Oh, yeah, faces. Mona Lisa there, it demonstrates a very simple principle of invariance. Faces as objects are invariant under rotation. You can just turn it upside down. That's an Euclidean transformation. But the interpretation of the facial expression
Yeah.
in this face is orientation dependent. The Mona Lisa picture has been manipulated a lot with the sad eyes and not a very charming mouth, which we didn't, perhaps, let's take it as given, which we didn't recognize on the original image upside down.
Yeah.
So
Yeah.
the
Eyes are right side up and the mouth is right side up.
the interpretation is something different. It's orientation dependent, whereas object recognition is not. There is a kind of invariance principle behind this issue too. Now, finally, I would go one step further, and we have an invariance principle in neurodynamics, which then eventually leads me to the following part two next week, if you have some time. And talk about the lens and mirror metaphor in neurosignaling issues. And that goes back with my work quite a while as well. Okay. I try to analyze neural situations in one or the other ways by computer simulation. But at the time, I have to add, I hardly had any computers available. We, we worked very roughly with the first huge desktops, and so with hardly any processing capability. But the idea, I think it's quite important, and discuss some of these principles even um, with different neuroscientists meetings at the time was quite interesting, particularly with Carl Pribram. Anyway, now in this picture, the idea is loosely sketched out. There's a source of signals, like from a nerve cell in the blue and the red lines here, axonal output of a neuron. The axon, also there's just one axon per cell, but the axons quickly arborize into an enormously complicated tree when they start to innervate neighboring cells. It's hardly possible to follow these connections, even among two cells. But the assumption was that, this cell communicates with another cell in a way that the feedback from the other cell has a certain meaning. It's a kind of optimized, maximized meaning. And I'm using the lens metaphor here. It's like you to spread out light through a lens, and the lens focuses it back to a point. And when you shift this point back to the cell, in the form of a feedback relation, you get this lens effect, and you simulate the lens effect. Now, if you program it and if you can test it, you can kind of study what comes out. The essential difference to at the time to computational models in neurobiology was that it does not just build on the action potential as such as all or nothing signals, it's binary signals, but it gives the action potentials, similar in the Hopfield version of it, a temporal characteristic, an interspike interval. So the time is important. The signal spreads out like a traveling watch. It changes the phase, it changes its time, the longer the path is. And it has got a small change if the path is short. The lens actually inhibits, in the middle of the lens, inhibits the change of phase until the signals from the periphery come down so that we can focus it. It's the metaphor is light, the propagation of light. I was then, of course, influenced by Richard Feynman's work. For instance, the idea came up, inspired by his work on the strange theory of light and matter, how photons travel through lenses or mirrors, or in space-time anyway. Inspired also of Carl Bribram's work about brain and perception, and also about David Bohm's work on his change of implicate and explicate order. Now, studying these signals, the question basically was, do action potential as all or nothing or binary signals, as was taken for the traditional metaphor, the brain is a computer, if you remember Churchland's work at this time. Does the action potential train transform to frequency or reciprocal space? So the lens effect in the brain that Carl Bribram suggested very much. And see what comes up. Now, for this, I developed a program for temporal coding in neurons and considering the neurons as a kind of coincidence detectors that was also emergent at the time. We published some of these details in this paper, Neural Face-Time Coding, and quantum neurodynamics. The point is, if you give the signals an interspike interval given phase property, then you can express the sequence of a spike pattern that was normally recorded in electrophysiology in time into a single vector, because the vector builds up by a vectorial addition, changes its orientation depending on the preceding time interval. So it changes a lot if the time interval is large, and it changes little if the time interval is small. And eventually, some of these vectorial phase diagrams provide a single vectorial magnitude that has a modulus, a length of the vector, and an orientation. But with that simple vector, you can actually express a whole spike train over time. And that was an advantage that we could apply to several situations quite well, actually. Now, I applied it, for instance, to this kind of brain mapping. It was the ISO orientation maps from area 18 of visual cortex. But the maps were not from me. The maps are from, at the time, came up in the 90s very quickly, for instance, from the Bonhoeffer group in Germany. Simply because that's invasive studies of in that case, of a cat cortex. And I never wanted and did not engage in invasive animal experimentations. Instead, but the maps were published, and I used them to test the performance of what I've just said about the phase coding and neurosignaling. The maps look a little bit like this. The cat is exposed to it's recorded from a orientation sensitive cortex, this layer there. It's area 18, and it's on the surface. It's recorded from layer 3 neurons on the surface, or mostly from, no, sorry, from layer 2 neurons, where the dendrites are spreading out in layer 2 of the 6 laminated cortex there. And, uh, and it's composed, of course, of the dendritic pattern of many cells. So one pixel encodes some about 100 neurons, one pixel here, because we had to reduce the resolution of the original images very much, simply because I had a very old computer, well, the computational performance was not very good. That was the limit of my desktop computer then. Anyway, but these are the original maps there under this orientation. So that means if the cat is exposed to horizontal orientation, it gives that picture. To that, if it's exposed to that orientation, it gives this picture. If to 90 degrees, it gives this picture, and that's 45 degrees, it would give this picture. And this is completely reproducible. So if the same window is recorded by high sensitive optophysiological measures with a camera that measures the distinction between, in principle, between the consumption or difference between hemoglobin and oxyhemoglobin patterns. So the consumption rate of oxygen in this layer. It's completely reproducible. It always comes up in the same way. So is there a code behind sensory maps? And is there an invariant behind the code of sensory maps? That was the question. At the time, codes behind motory maps were well around, also in behavioral studies. One prominent person was Georgopoulos, a Greece scientist in the US, who studied the performance of in the motory pattern that when the signals build up in the population vector, and the population vector reaches its maximum shortly before the animal, again, the subject, executes a certain motory program, like hinting in a certain direction. But there was nothing around about a possible code behind sensory uh, uh, coding, an analysis of the sensory codes. What's the idea behind this map? What do neurons actually do? Altogether, the window here comprises perhaps about 10,000 cells. And one pixel in my simulation encodes about 100 neurons there, the dendritic pattern there. Okay. Now, as a result, as a short result, I'm sorry for the quality of these diagrams, but at the time it didn't have better possibilities there. I think I cite this was here. Applying the system or the computer or the programs that I just mentioned there to these different orientations. We don't have to consider 108 degrees because it's just an orientation sensitive map, not a direction sensitive map. The population vector that comes up that I mentioned before as a result of that simulation, the population vector starts to increase between 0 and 90 and 45 degrees. So in a p quarter, and then goes down again to p half issue. And it starts doing the same between 90 degrees and 180 degrees again, coming back to the same situation. This is the size of the population vector. The population vector changes length just within a quadrant of that orientation map. So the neurons only have to decode a quadrant, not the entire P orientation, because then they just change sign, if you want, they are reversed and they start doing the same again. That's one of the principles how a certain patch in the neural and the cortex can encode so many different things at once because it's a parsimonious solution for it. And the parsimony comes up with that point that the map for zero, with this orientation as a stimulus, is a kind of inversion for the map at 90 degrees. On the way from this orientation to 90 degrees to p half rotation, the population vector encodes the situation, but then it can start off again from its inversion. So as an inversion is a mirror image in the form of intensities in 3 dimensions, if you imagine this in 3 dimensions. And it plays a role. It's there. Now, the final issue for today, just a couple of minutes. What I did here is just to show again that there is something like an inversion or a reflection or a mirror image detectable in the coding principle behind sensory maps. That's how I would summarize it. Okay. Now let's proceed to the next level that we mentioned, from simulation from classical to quantum at the molecular level, basically. When I look at the molecules that are mostly engaging in the signaling pattern in the brain are the ion channels and membranes. One of the findings was that in atomic force microscopy, I'm citing this study here, was that the shape and the length of these oriented molecules inside the membrane, these are ion channels, particular potassium ion channels there, actually changes with the state of the molecules. And the state of the molecules here is compared between a closed gating state and an open gate state. In the open gate state, the molecules is conducting between the intracellular and extracellular side. And in the closed state, the ions are at least not available for conduction. And there is enormous difference in the shape of these molecules depending on the state of their conducting, or let's say, availability of ions for conduction. So the invariance principle, I'm just taking this as an example there, shape plays a role all of a sudden, where before at the macroscopic scale, at the global anatomical scale, I just said shape doesn't play a role, or not essentially. But topological properties play a role. But here we are back again. Invariance principles are different at this molecular and atomic level. Shape, length, and distances start to play a role again. Naturally, quite naturally, because all allosteric interaction between these molecules greatly depend on, for instance, Coulombic type of interaction between charges. And they are, as we all know, dependent on distance. Close approaches or far distance, and etc. So at this level, things change again. And there are more changes between the global macroscopic level and the molecular level than this one. Now we studied some of, I'm giving a picture here with one of the influential persons there. That was Massimo Cocchia. unfortunately died as well a couple of years ago. And the studies I'm just mentioning greatly involved Johann, who unfortunately is not here today. Hopefully next time, and we have more on Johann's work,
He's here.
Johann Somhammer. We studied the role of shape and distances in a very small subdomain of these conducting proteins of ion channels in the selectivity filter. The selectivity filter, which is strongly responsible for conduction as well as for specificity to particular sets of ions. And we found out that you cannot distort the selectivity filter from its position. Whatever you do is whether you shorten it or whether you press it, the conduction almost very quickly diminishes or abolishes. It can abolish completely. So shape and length and distances start to play an important role, and the invariants are different than on the global scale. Now, concluding this part one, I think I'm precisely in time, surprisingly. Concluding this part one, can I transfer it again? Roughly, because I will come back perhaps in part two again there. I would say that I mentioned the empiricist approach as an epistemic approach to consciousness. The empiricist route is to set out from sensory perception that leads to conceptual space, to ideas, concepts, thoughts. They're not a kind of spirits that we will receive from some spiritual world. We actually produce them ourselves. Maybe a little bit of a radical point of view, and then of course many philosophies would argue against this, but I'm convinced that this is the case. And somehow we under and overestimate our own abilities. We underestimate it when we think about that we are just receivers of ideas in the Platonic sense, and we kind of overestimate our issues as well. On this route, the concept of invariance principles helps us, as I've tried to show, to dissociate scale-dependent changes of organizations. We have seen that the invariance principles at the global scale, the total brain scale, are different from the molecular scale, and we'll find out more about this. At the macroscopic level, topological properties like numbers and handedness are dominating. Handedness is there. Left and right hemisphere differences have been reported all the way through. If you think about the difference between the two hemispheres, Roger Sperry has received a Nobel Prize I think in the 60s or so, about the split brain process. But numbers are more important than volumes and shapes. Psychological invariance, concept different than invariance properties. Object recognition is invariant under orientation, reversing transformation, whereas the interpretation of meaning is not. So it's a kind of the mental issue that comes up from an organization is completely different somehow, but the relation, it all points to the way that the relation is somehow mirror. Well, I will come back to this point. At the molecular level, Euclidean principles dominate, as it just said. Affine transformation do not render functional invariance. But the orientation reversing organization, so heterochirals, for instance, do not exist. does not exist in the important molecules that we have, and in polynucleotides, RNA, DNA, does not exist in proteins. It does not exist in sugars. It does not, or partly, does not exist in lipids. So we have strict homochirality. And I will use this kind of and interpret this homochirality then in the following part two a lot. Okay, thanks for your attention. The next part will go into more details.